Is this microphone broadcasting? We good? Anybody not hear me? Okay, I'll take that as no objections. So obviously I am not Kristina. For whatever reason, she couldn't make it, so my name is Shannon. I will be covering her session today, so fear not, it will still happen. I do not normally SI for genetics. I am normally the Bio 1 SARC tutor, so bear with me, I do not know everything. But we're just going to be going over what Kristina had planned for you. We're going to do her practice exam that she made. That's pretty much it. So this exam we're going over that Kristina made for you guys is on this website here. So don't worry about copying down any questions. Everything's there online. Just make sure you get the answers before you leave. So that's what we're going to be doing. If you don't have this link, write it down now. I'm going to take it off and then we'll get started. Everybody good? We got the link? Cool. All right. Number 1, which DNA sequences are always identical? They're going to be only our sister chromatids. Homologous pairs could potentially be identical, but more likely than not, they're not going to be identical. The only situation where our homologous pairs could be identical is if every single gene on both of those chromosomes had exactly the same alleles. And I imagine the chances of that would probably be really slim. So the DNA sequences that are always going to be identical are going to be our sister chromatids. Because remember, when we make our sister chromatids, we are making a direct copy of the chromosome we already have. When we have homologous pairs, we got one chromosome from mom and one chromosome from dad. And those could have totally different alleles. Our homologous chromosomes are both going to code for the same traits, but those traits could have different alleles. So that would be why homologous pairs would not always be identical. And see, parent cells and their fertilized offspring, if the offspring are fertilized, that means they got genes from mom and from dad. And if they got genes from mom and dad, they're not going to be identical. So the only answer that could be right would be A, sister chromatids. Number 2, growth occurs during what phase or phases of the cell cycle? The only cell cycle phases that we have growth are G1 and G2. S phase, what are we doing? Synthesis. In S phase, we're doubling all of our DNA. We're going through DNA replication. So there's not really any cell growth going on there. What's happening in M phase? Actual mitosis, yeah. So this is going to be our mitotic phase. And then what's G0? I can't hear what anybody's saying. It's our non-dividing phase. So if we're non-dividing, we're not going to be worrying about any growth. So think of G0 as like nerve cells. Nerve cells rarely, if ever, will go through cell division. So nerve cells are almost always going to be in G0 phase because they are not dividing. Any of the other phases, G1, G2, S, or M, would indicate that the cell is dividing. So the only time we're going through growth is going to be G1 and G2. All right, number 3. A cell in G2 phase has 16 chromosomes. We want to find out how many DNA molecules are in metaphase. So if we're in G2 we're going to have 16 replicated chromosomes. That means we're going to have 16 X's. That means each X, or each of the 16 chromosomes, is going to be made up of 2 DNA molecules. Nothing different has happened between G2 and metaphase. So that means we're going to have 32 DNA molecules. So we have to understand that since we're past S phase, every chromosome is going to have 2 DNA molecules each. So if we're in metaphase, we are still replicated at this point. So all of our DNA, or all of our chromosomes, will have 2 DNA molecules. If we were in anaphase, we would not have 2 DNA molecules per chromosome. Because remember, in anaphase, we're pulling apart our X's, our replicated chromosomes. And they're going to be 32 unreplicated chromosomes, each made of 1 DNA molecule. Everybody good? All right, number 4. During what phase does crossing over occur? Prophase 1, always. Crossing over is always going to occur in prophase 1 of meiosis. Do we have crossing over at all in mitosis? No. So we could already get rid of the 2 mitosis choices. And it's not going to be interphase. So pretty much we just have to remember which prophase of meiosis crossing over is going to happen in. And that's going to be prophase 1. So more really just a fact you need to memorize. Crossing over, prophase 1. All right, number 5. Chiasmata are, this is going to be D, areas where crossing over occurs. What is A, proteins located at the center of the chromosome where the kinetochore will form? What is that? That's going to be the centromere. I don't even think B is a thing, so it's more of like a bogus answer choice. Areas of connectivity between sister chromatids, that's also going to be a centromere, but another way of stating it. Areas of difference between sister chromatids, I don't even know what that means. So that leads us to D. Crossing over is going to occur at the chiasmata. All right, number 6. When does the degradation of Shugoshin occur? So this would be after meiosis 1. So Shugoshin, if I'm pronouncing that correctly, is something that helps prevent degradation of centromeres. We need the Shugoshin to keep our sister chromatids attached. So we want to make sure during the whole process of meiosis 1 that our sister chromatids do not come apart. Because remember, in meiosis 1, we are dealing with pulling apart homologous pairs, not sister chromatids. So we want to make sure that Shugoshin is active the whole time in meiosis 1. Because in meiosis 2, that is when we are actually pulling apart sister chromatids. If we're ready to pull out those sister chromatids, we don't need that anymore. We don't need the Shugoshin anymore. So we're going to get rid of the Sugoshin after meiosis 1 is complete. Okay? If we got rid of it before that, we'd have problems, because our sister chromatids start separating before they should. And we don't want it to happen too late, because then our sister chromatids wouldn't separate in time. So we want to get rid of the Sugoshin after meiosis 1. All right, number 7. Choose the correct statement. Okay, let's go through these. So A, meiosis 1 is a reductive division that produces 4 haploid cells, while meiosis 2 is an equational division that produces 4 diploid cells. So there's a lot of things wrong with this. First of all, we do not produce 4 haploid cells in meiosis 1. And even if you missed that and you kept going, we know we never make diploid cells in meiosis. So that tells us it's not A. B, meiosis 1 is a reductive division that produces 2 haploid cells. That's good. It is reductive, and we do produce 2. While meiosis 2 is an equational division that produces 4 haploid cells. So that looks good so far, but let's go through the other choices just to make sure something's not more correct. So C, meiosis 1 is a reductive division that produces 2 haploid cells, while meiosis 2 is a reductive division that produces 4 haploid cells. So this would not be correct because meiosis 2 is not reductive. Reductive means we go from having, let's say we started at diploid and then we went to haploid. We're cutting our number of chromosomes in half. We do not do that between meiosis 1 and meiosis 2. Between meiosis 1 and meiosis 2, we're going to be equational. We have the same ploidy through both stages. So that would make C incorrect. D, meiosis 1 is equational division that produces 4 haploid cells, while meiosis 2 is an equational division that produces 4 diploid cells. So there's a lot of things wrong with this. First of all, meiosis 1 is not equational. It would be reductive, because in meiosis 1 we become haploid, so that wouldn't be right. And we also know that we make 2 haploid cells at the beginning of meiosis 1, not 4. Also, meiosis 2 is equational, and it does produce 4 diploid cells, so the second half is good. E, meiosis 1 is equational division that produces 2 haploid cells, while meiosis 2 is an equational division that produces 4. So the only thing wrong here is this word here, equational. So it is not equational, it is reductive. And that means B is the correct answer. So meiosis 1 is reductive because we go from being diploid to haploid, and we make 2. Meiosis 2 is equational because our ploidy stays the same, and we make 4 daughter cells that are haploid. Good? Question?
So how is it in meiosis 2 it's equational if the sister chromatids are being pulled apart?
It's equational because the sister chromatids are being pulled apart. Think of what we do in mitosis. We start diploid and we end diploid. It's because we replicated all of our chromosomes, we pulled all our sister chromatids in half, and we put half of them in each cell. So we didn't divide the number of chromosomes in half, we divided the number of sister chromatids in half. Does that make sense? So think about in meiosis 2, we start off being haploid and we end haploid. So we didn't cut those number of chromosomes in half anymore.
Oh.
Make sense?
Okay, yeah.
Is everybody good with that? So equational just means the number of chromosomes stayed the same. Reductive means they got cut in half. Okay? Cool. All right, 8. Choose the statement that is most correct. So A, a genotype is a set of alleles, while a phenotype is the physical manifestation of those alleles. That sounds good to me. B, a genotype is a set of genes, while a phenotype is a physical manifestation of those genes. It also sounds good. C, a genotype is a set of genes, while a phenotype is a set of alleles. Okay, that's definitely not good, because phenotype is not a set of alleles. The other two were good in that phenotype is a physical manifestation, but one of these is a little bit better than the other, and that's going to be A. The genotype is going to be alleles and not just genes. So when you look at your specific genotype, you're going to be looking to see if you're homozygous or if you're heterozygous for a particular gene. So that's why the word allele here is a lot better than gene. When you look at your genotype, you want to see what particular alleles of that gene you have. You're not just looking at, oh, my genotype is that I have a gene for hair color. We want to go more specific and say, what is that allele for the gene hair color? So that's why A would be more correct than B. So be careful with wording. It's a little tricky. All right, number 9. An allele is defined as A, a form of a gene found at a given locus. Could be. B, a set of nitrogenous bases coding for a functional product. Right away, I know that's not right, because a nitrogenous base would be something like A, T, C, G, or U. So that's not our answer. C, the location of a gene along a chromosome. That wouldn't be allele. That would be our locus. D, the site along a gene for RNA polymerase binding. That's definitely not an allele. That would be something like a promoter. So it's not that. And the termination point along the gene, that would be something like a terminator. So that leaves us with A, a form of a gene found at a given locus. And we kind of already mentioned that in the last question. We know that an allele is a specific version of a gene. So if my gene was hair color, my allele would be red hair. So it is a specific form of a gene. All right, number 10. Mendel's first law, the principle of segregation, states that alleles located at different loci segregate independently. Is this true or false? It's a little tricky. This would be false, because what law states that alleles are going to separate independently? Law of independent assortment. So I feel like those two hypotheses are really close, and it's kind of easy to get them confused. So be careful in that you know the difference between those two laws.
So what is the first law?
The first law, I don't know which one's actually first, but the principle of segregation is not the correct name. So I'm assuming the first law would be the law of independent assortment. The principle of segregation is saying when we make a gamete, we either put one of our alleles or the other. Because remember, we're diploid organisms, so we have 2 alleles for every gene. And the law of segregation states that I'm going to put one of those 2 in every one of my gametes. The law of independent assortment is saying that regardless of what letter I'm dealing with, I'm going to put one in this gamete and one in that gamete regardless. It's completely random which one gets put in the gamete. So principle of segregation is saying that we're putting one in each gamete. Law of independent assortment is telling us that it's independent, which one we put in there. So they're very close, so be careful with that. And know the definitions of each. Okay, number 11. A geneticist crosses a set of pea plants, paying attention to height. He examines the genotypes of the offspring and finds 1/4 are homozygous dominant, 1/2 are heterozygous, and 1/4 are homozygous recessive, where big T is the dominant tall allele and little t is the recessive short allele. What percentage of the offspring is tall, and what probability rule was used to reach this answer? So we know that 1/4 are big T are homozygous dominant, and we also know that 1/2 are heterozygous. We really don't even need the information for homozygous recessive because they're asking us for tall, the tall phenotype. So we know we're going to use the addition rule. So I can already get rid of C, D, and E. We're going to use the addition rule because they already gave us all the information we need. We don't need to do any multiplying. We're not finding the probability of this and the probability of that or anything like that. We already have everything we need right here. We have the probability of one, we have the probability of the other, so all we're going to do is add them. So if I have 1/4, and then 1/2 is the same thing as 2/4, that gives us 3/4. So 11 would be B, 75%. So all I did was take probability for being tall and combined them. It's just that they gave us a little extra information, and they gave us probabilities based on specific genotypes, whereas all we needed to do was calculate it for the phenotype. Everyone good? Everybody got this? Okay. All right, number 12. A YO genotype is indicative of. So first of all, we know YO, I don't even know if YO could exist because you don't have an X chromosome. So right away, we know it's going to be lethal. It's just not possible to survive without an X chromosome. Remember that there's a lot of sex-linked diseases because the Y chromosome doesn't carry it. So if we had no X and only a Y, we just would not have enough genes for everything. Question?
How beneficial do you think it would be for us to know, be able to identify all of those, concepts?
I would probably know them all. We'll go through them. But yeah, I think it would be important to know them. But yeah, if you're missing an X chromosome, it wouldn't work. It would just be lethal. Because we're missing such a big amount of DNA in that pair of chromosomes that it just wouldn't be able to function. So let's go through the other. So if we have Klinefelters, what is our genotype for the sex chromosomes? Anybody remember? Yes, XXY. So you are a male with an extra X chromosome. If we have triple O X syndrome, what does that sound like it means? 3 X's. I think they also call it triple X, but it's pretty self-explanatory. It just means we have an extra X giving us 3 total. If we have Turner syndrome, what would our genotype be? Right. So usually they designate it as XO. It will be a female that is missing an X. So similar to the YO genotype, but because we actually have an X, we can still function perfectly fine. So XO is a perfectly viable genotype, whereas YO is not. And then finally, a super male, XYY, so a male with an extra Y chromosome. I don't know the specific implications of having each of these. That might be important to know. I don't know them off the top of my head, but at the very least, I would definitely know what genotype constitutes each of these syndromes. All right, number 13. Under genic sex determination. So first of all, when we're talking about genic sex determination, we are talking about, this would definitely not be in humans. We're talking about a situation where we don't really use the shape of our sex chromosomes to determine anything. So based on that, let's look at our answer choices. So A, sexual phenotype develops over time depending on environmental conditions. So no, it's not really dependent on environmental conditions. B, sexual phenotype is present at birth depending on conditions during development. No, that wouldn't be right either. C, sexual phenotype is determined by a combination of sex chromosomes present. So no, that would not be genic. That would be what we do and use chromosomal sex determination. So that would be like in humans, but that's not what the question is asking. D, sexual phenotype is based on genes, but chromosomes appear the same across sexes. So that sounds good, but let's read the last one just in case. E, sexual phenotype is not based on genes, but sex chromosomes still appear the same across sexes. So kind of tempted to pick that one, but no, that would not be it. Our correct answer would be D, because in genic sex determination, it's our actual genes that are determining what sex you're going to be, and the chromosomes are exactly the same. They're not like in humans where we have an X and a Y. They're going to be identical. And it's going to be like some gene on the chromosomes that determine the sex. So that would be genic sex determination. Different than what humans do, which would be chromosomal sex determination. Question?
So you're saying, so chromosomal in the sense that if you have two X's, you're a girl, two X's up.
XY or male, yeah. Where in this case, it's not like XY that determines it. It's the genes on whatever the sex chromosomes are. They're not going to be different.
So both sexes would have like the same.
Same exact chromosomes. Yeah, they just have different genes, and that will determine what the sex actually is. All right, 14. In a group of 30 turtles, 10 have no green pigment. 12 have a lot of pigment. 5 have a moderate amount of pigment. And 3 have very little. This is an example of what? So, expressivity is displaying different levels of that trait, different levels being displayed. And penetrance is pretty much if you display the trait or not. Display trait. So in this case, it's going to be both variable expressivity and variable penetrance. And it's going to be that because we have turtles that have no pigment, and we have turtles that have pigment. So for that gene, we could have no pigment or pigment. That would be our penetrance. And because we have different levels of displaying the green pigment, so we could have a lot, a little, or a moderate amount of the green pigment, that's going to be our variable expressivity. So that's going to make E our correct answer choice. If we were in complete dominance, we would see something like, I'm going to use a different color so it makes it easier. If we had red turtles and white turtles and then pink turtles. Incomplete dominance means one doesn't completely mask the other. But because we have such a range, that wouldn't just be incomplete dominance. If we were talking about codominance, that would be if our turtles were displaying multiple colors on their shell. Codominance is when both alleles would be dominant. So I'm going to use a different color because I think it makes it easier also. I'm going to use the pink example again. If we had red turtles and we had white turtles and it was codominant, we'd also have turtles with spots of white and spots of red. They would just be showing both alleles. Does that make sense? So that's why neither of those cases would be in this example, because we have so much different variation going on. All right, 15. Which forms of inheritance appear approximately equal in males and females? So pretty much we want to look for something that has nothing to do with sex-linked or anything related to that. So right off the bat, we know for sure it's going to have to be A and B. Autosomal recessive and autosomal dominant have nothing to do with what sex you are. So that's going to happen equally in females and males. The other three wouldn't. So whether it's recessive or dominant, doesn't matter. As long as it's sex-linked, we're going to see different percentages in males and females. And if you were to do a Punnett square for something like that, you would see why. Because remember, when something is X-linked, the Y chromosome does not carry an allele for that trait. So if a Y chromosome doesn't carry it, we're going to have a disproportionate number of alleles going through generations. So it's not going to be X-linked recessive or X-linked dominant. And E, let's see. I wrote this one down. Yeah. No, I did not. I don't remember exactly what holandric is. Does anybody know off the top of their head? Oh, okay, that's Y-linked. So, wait, am I reading two different questions? I didn't realize this was twice. Okay, this is Y-linked. So yeah, if something is Y-linked, it's also some sort of sex-linked going on there, so it's not going to be equal in males and females. Okay, I got ahead of myself. This is Y-linked. All right, 16. Which form of inheritance skips generation? So it's going to be the recessive traits that skip generations. If something is dominant, I mean, straight off the bat, because it's dominant, it makes it seem like you're going to see it. So if something is dominant, you're going to see it in every generation. It's really difficult to mask a dominant allele. I mean, the only way we could do that is if something was homozygous recessive. So the ones that are going to be more likely to skip generations are going to be anything recessive. Oh, somebody should have told me you couldn't see it. You guys can yell at me. Does anybody know if holandric is, it's Y-linked, but is it recessive, dominant? It's what? I mean, that's the extent of my knowledge on whatever that means, so. I know that does not skip generations, so that's all I can tell you about that. All right, 17. In the case of complete linkage with no crossing over, the progeny of a heterozygous and homozygote R This one is going to be E. Because, let's see, there's going to be complete linkage and no crossing over. So if there's no crossing over and our genes are completely linked, that means none of our DNA is going to be new. We're not making any new combinations. So that's why it is non-recombinant. And we're going to have a parental phenotype ratio of 1 to 1. So we can get rid of A and B and C, because we know they're all going to be non-recombinant, because there's no crossing over. Everybody good? (18) During crossing over, which DNA molecules swap segments? So our sister chromatids, they never swap with each other. They're going to be direct, identical copies of each other, so it's not that. And then pretty much we have to determine between B and C. It's not going to be both. And since we crossed off A, it's not going to be all of the above. So we know crossing over happens between non-sister molecules. But we need to figure out, does it happen with the same homologous pair or a different homologous pair? It's going to be the same, right? It's going to be the same homologous pair because remember when we have crossing over, we have those two chromosomes lined up, and this makes up our homologous pair. And when we have crossing over, it's going to be between the chromatids that are connected between the two chromosomes in the homologous pair. If we had another homologous pair over here, so this is a different homologous pair, they're swapping segments with themselves. Nothing happens between the two different homologous pairs. So that's why it would be B. We never have any DNA swapping with different homologous pairs. If we did that, we would mess everything up. Because homologous pairs, both chromosomes are going to have genes with the same trait. So let's say right here is a gene for hair color. If I swapped that with this homologous pair, which could have completely different, let's say right here on this chromosome is eye color. If we were to swap segments with a different homologous pair, we'd be swapping segments with completely different genes. All we want to do with crossing over is get different alleles to mix up our genes. Everybody good? So we're always going to swap with the same homologous pair, non-sister chromatids. Okay, 19. I will be perfectly honest and tell you I don't know how to do this problem. What I can tell you is give you some information and help you guys, and maybe you can help me to figure out the rest. So they tell us we have 1,000 flowers. 650 are red, and 350 are white. And we want to calculate allele frequencies of both of the alleles. So what I do know is that total, we're going to have 2,000 alleles. And we know that of those 2,000 alleles, 700 are white. So that means we're going to have 700 of the R, of the little r. But the one thing that is catching me on this problem is we have 650 red. That means 650 are either homozygous dominant or heterozygous. Does anybody know how I'd figure out the rest? Okay. Mm-hmm. Mm-hmm. So
Yeah, P is your dominant, Q is the recessive. Yeah.
right. So, I mean, technically we have the little r, we have Q, but we would use the first, so we don't even need this. So our Q would be the 700 out of 2,000, right? Does that math to, let me see. So if we take, so I know the right answer is A. So if 700 out of 2,000, so yeah, that's where I'm confused. We get 0.35, which would, Q would be the percentage of little r, right? The square root of it? Oh, I see. Okay, that would make sense. So we are using this formula. P squared plus 2pq plus q squared equals 1. So we found q squared. Okay. So is p plus q, is that for genotypes? And the second formula is for alleles? Is that right? No? So if we're trying to find allele frequencies, why would we use the second equation? Do you know? So we can't even use this formula? Yeah, because these are the Hardy-Weinberg formulas. So I don't know what to tell you guys. Hardy-Weinberg is something I have not looked at for a long time, so you have to forgive me. Although I can tell you, to get this answer, we did the 700 divided by 2,000, and we took the square root. So hopefully this is enough information for you guys to work it out on your own, or you'll have to ask somebody else. But I do not know. I apologize. And then, of course, if we have the r, that would be our q in this formula, and we can subtract it from 1 to get big R. I don't think so. The only reason we take the square root if we did use this formula. But I'm not 100% sure. And it doesn't tell us that it's in Hardy-Weinberg equilibrium either, so I don't even know if we're allowed to make the assumption that we can use that equation. So yeah, I'm sorry, but A is the right answer, so if you're able to figure it out on your own, then that would be good. But I'm sorry I can't help you guys. All right, moving on. All right, 20. What statement is true regarding the structure of DNA? So for this one, it is C. I think this is just some fact you need to know. So the 9th nitrogen of purines attaches to the first carbon of the deoxyribose, which is the DNA sugar. I'm not sure how you'd figure it out besides just knowing it. All right, 21. A molecule of DNA has 32% cytosine. What percentage of uracil is found in the transcribed RNA? So if we have 32% C, we already know that we also have 32% G. C and D are complementary, so we're always going to have equal amounts of each. So that gives us 64% total. So that means we have 100 minus 64, which is 36. So that leaves us 36% left for A and U. So if we have 36% left, we're going to split it equally. And we're going to say that we have 18% A and 18% U, which is A. So the math is pretty simple. You just need to know for this problem that amount of A is going to equal the amount of U, and the amount of C is going to equal the amount of G, and they both add to 100%. Yes?
So if you would have just kept it as a molecule of DNA has 32% cytosine, what percentage of uracil is found? It's 0. Right? Yeah. Yeah. If you would just look at a molecule of DNA has 32% cytosine.
Oh, I see what you're saying. What percentage of uracil is found in the transcribed RNA? I mean, we're going from DNA to RNA.
No, but I'm just saying, like, if you took out the RNA part and it was literally just a molecule of DNA has 32% cytosine.
Oh, and it was asking for uracil? Yeah, it'd be 0. If it was asking for percentage uracil in DNA, yeah, it would be 0. Because we know that DNA doesn't have uracil, yeah. DNA has thymine instead, RNA has uracil, so yeah. Yeah, if they asked what percent uracil in DNA, it would be 0. Yeah, RNA you can calculate it because RNA does have uracil, whereas DNA does not. Okay, 22. Which of the following statements is correct regarding DNA replication? A. Only new strands on the leading strand are synthesized in the same direction as the movement of the replication fork. That sounds pretty good, but let's read the other choices. B. Both new strands are synthesized in the direction of the replication fork. All right, no, that's not true. C. There is only one origin of replication, just like in bacteria. That is also not true. We know in DNA replication, we're going to have many, many points of origin because we're going to have those bubbles of replication. D. DNA is synthesized in the 5 to 3 direction on the lagging strand, but the 3 to 5 direction on the leading strand. That is not true. DNA, no matter leading or lagging, is always 5' to 3'. No exceptions. Anytime we're making new DNA or new RNA, we make it from 5' to 3'. So D is not right. And E, the lagging strand is also known as the sense strand. No, that is not true. The leading or lagging could be the sense strand. Sense strand just means it is going to be the template strand for our transcription. So when we transcribe the template strand in transcription, it's going to be the sense strand. So that doesn't matter. It has something to do with being leading or lagging. So that means A is right. Only new strands on the leading strands are synthesized in the same direction as the movement of replication fork. So that's pretty much the basic definition of being the leading strand. The leading strand is the leading strand because 5' to 3' is going the direction that the replication fork is opening. If we're lagging, 5' to 3' is going the opposite direction. So if I draw that real quick, just so we can see, if this is 3, 5, and my new DNA is being made from 5 to 3, and let's say our DNA is opening this way. If 5 to 3 is going the same direction as the strands are being opened, this is the leading strand. Because this strand has to go this way, because this is 5 and that is 3, that makes this the lagging strand. So if it's a leading strand, 5 to 3 goes in the same direction that the DNA is being opened. If it's the lagging strand, 5' to 3' is going the opposite direction that the DNA is being opened. Does that make sense? Are we good with that? I'll assume your blank stares mean we're good with that. right, 23. Which enzyme involved in DNA replication has proofreading capabilities? Oh, sorry, you have a question?
So that's happening on one side of the replication force?
Yes.
Like, what's happening on the other side?
The other side is just going to be the complete reverse. So if in this case the top is the leading, the other half is going to be lagging. I can draw that real quick for you, just so it's clear. So yeah, I only drew half, but that was bad. So this is my whole replication bubble. You kind of divide a point here. So if this is 5 and this is 3, this is the 3 and that's the 5. So that's 3, this is 5, this is 3. So this is going to be like our origin in the middle here. So our new strand would be going this way from 5' to 3'. So this direction, and the strands are opening this way. These strands are opening that way. So 5, 3. This one's going this way. But if we were on this strand, we would be, this one would also, they're going to be going the same direction. But this time, since the opening is this way, that makes this the lagging strand. So, oh, I'm sorry, this is the leading. Because this is lagging, this is leading, so this is lagging. Because this is still going from 5 to 3. Does that make sense? So it's always going to be going this direction because that's the 5 to 3' direction. And this one's always going to be going 5 to 3 this way. It just depends on at what point are we going the same direction as the opening and what point are we not going the same direction. Everybody good with this? I know sometimes the leading and lagging strand can be a little confusing. And I usually think it's easier to think of half of the replication bubble at a time rather than the whole one. So if you get confused, think of just half of it. But if we see that 5 to 3 is in the same direction as the opening, it's a leading strand. If 5 to 3 is in the opposite direction of the opening, it's a lagging strand. But always, 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 new DNA made from 5 to 3. Is that good? Okay, cool. Everybody good with this picture? I'm going to move it. All right, back to 23. Which enzyme involved in DNA replication has proofreading capabilities? So, pretty straightforward. This one is going to be B, DNA polymerase 3. So DNA polymerase 3 is in charge of actually making the new DNA strands. So they're going to add the nucleotides. And while it does that, it can kind of proofread as it's going along. I know specifically DNA polymerase 2 can come in and proofread, but that would be as like an added proofreading to what DNA polymerase 3 has already done. So we have a lot of enzymes that would proofread because we want to make sure this process is going to be perfect. So DNA polymerase 3, while it's adding the nucleotides, can also proofread. What does DNA polymerase 1 do? I think you're thinking of something else. Anybody know? I don't know what specific enzyme that one would be. Well, DNA polymerase 1 is going to be the one that removes the primers. It removes the RNA primers and replaces with DNA. Does that ring a bell? And what does ligase do? Connects to Okazaki fragments. What does primase do? It makes the RNA primer. And what does dry erase do? Prevents overwinding. So cool. We got a review of some of the other enzymes we use in DNA replication also. All right, 24. Which statement best describes the transcription apparatus most commonly seen in bacteria? So this one is going to be C. 5 polypeptides and a sigma factor form a functional holoenzyme. So I think this is probably purely memorization and knowing how many polypeptides and what form what kind of enzyme. So in this case, we're going to make a holoenzyme. And we know that to do that, we have to have a sigma factor and 5 polypeptides. Okay? And those are going to be the ones that are the common transcription apparatus in bacteria. All right, 25. Which statement is incorrect regarding row-dependent termination? So this one is going to be B. Row-dependent termination does not involve a rut site, I can't talk, downstream of the termination sequence. But RNA polymerase does pause at a terminator sequence. It does occur in bacteria. A hairpin is formed in the RNA molecule. And it does end with the unwinding of the RNA molecule from the DNA template. So one incorrect is B. Yes?
It involves a rut site upstream? Is that what that means?
I don't know, to be honest. I'm not 100% sure. I would double check that. Yes. Independent does? Okay. So this would be for row independent. Thank you for that. All right, 26. What is the function of the RAT1 protein? This one is going to be C. It operates in eukaryotic termination. '27. What is the function of the 5' cap? This one is going to be C. We know this one isn't A because we know 5' caps are something we're going to find on mRNA. So it has something to do with tRNA stability. It's not going to transport all these other types of RNA because we know it only deals with mRNA. And so obviously it's not all of the above or A and C, but it will be C. because the 5' cap is what the mRNA has on it so that the ribosomal subunit recognizes that it's mRNA and that it's ready to start translation. So keep in mind, we got our 5' cap to our mRNA, and then from there we can figure out that the answer had something to do with ribosome and translation. To what? I believe this would happen in eukaryotes as well. Because I know in eukaryotes we still have to add the 5' cap so it could safely leave the nucleus. I'm not 100% sure, but I would assume that would also be the case in prokaryotes. They don't have a 5 cap? If they have a guanine cap? They have a guanine cap in prokaryotes? No, no guanine cap. So they have a 5' cap, but it's a little different? Do they have the poly A tail? So prokaryotes have something similar. Is that right? Prokaryotes have something similar to this? Okay. Because I would imagine they would still need something so that the ribosome could find it. Yeah. Okay, so yeah. So something similar will happen in prokaryotes, but they don't have a 5' cap exactly. So then this would specifically be eukaryotic. Okay, 28. Which type of enzyme functions in eukaryotic transcription? So our first key here is that we're talking about transcription. So right away we can get rid of D and E because we know we're going to use some kind of RNA polymerase in transcription. The only time we use DNA polymerases is in DNA replication. If we want to make mRNA, we want to use an RNA polymerase. And the specific one that we use in this case is RNA polymerase 2. From what I know, RNA polymerase 1, I believe it only makes rRNA. And I'm not 100% sure what RNA polymerase 3 does. What? It's for what? tRNA. Okay, thank you. Cool. So this one is for tRNA. So that means this one has to do with mRNA, because remember, in transcription, we're making our mRNA transcript. I'm not 100% sure. I know it produces some kind of rRNA. I don't know what specifically it does. Yeah. But what's important to know is RNA polymerase 2 is the one that's actually making our mRNA transcript in transcription. Okay, 29. The 3' end of a transfer RNA molecule contains the sequence. Anybody know? CCA. Yep. I don't know what all of these are, but another one that could be tempting is AUG. Everybody knows AUG. What is it? Start codon. I don't know the others off the top of my head. Are any of the other ones significant at all? Anybody know? I feel like some of them could be stop codons, so I'm not 100% sure. Is B a stop codon? No? Okay. I don't know if they're anything significant, but A would be the answer. That's going to be the sequence on the end of the tRNA. The only one, to me, that kind of threw me off was D, because I know AUG is also significant. Of course, it's on mRNA, not tRNA, though, so that's what led to getting the right answer. All right, number 30. Why do we consider the genetic code to be redundant? So A, only contains as many codons as there are amino acids. That's not true. We know that there's a whole lot more codons than there are amino acids, because amino acids could be coded for by multiple codons. B, contains more amino acids than there are codons. That is also not true. We just said the opposite, that there's more codons than there are amino acids. C, single tRNA molecules may attach to multiple different amino acids. I'm not 100% sure if that's true. Let's keep going. D, there is only one start codon but multiple stop codons. That's true, but that doesn't really have anything to do with why the genetic code is redundant, so I'm going to cross that one out. And E, amino acids may be represented by multiple codons. That's definitely the answer we're looking for. That would explain why there's redundancy in this. Because you could have several codons, and they could all code for the same amino acid. Okay, then yeah, that makes sense. So then it wouldn't be C either. That was the only one that threw me off a little bit. Okay, 31. In eukaryotes, where is the start codon found? Do we know? A, Kozak sequence. Where would the Shine-Delgarno sequence be? That's in prokaryotes. Yep. And then initiator, promoter, and enhancer all have to do with what? Transcription. So if they're specifically asking us for start codons, that's indicating to us that we are in translation. So the only things that relate to translation, this would be A and B. C, D, and E are all transcription things. And then we just have to go a little further and know that in eukaryotic, it would be the Kozak sequence. Shine-Delgarno would be in prokaryotes. (32) IF3 has what function in translation? This one is going to be C. So when we use IF3, we use it because we need to make sure that the first tRNA can get in our ribosome before the actual ribosome, ribosome subunit forms around our mRNA. So that's why it would be C, prevents the binding of the large and small ribosome subunits during initiation. So we want to make sure our tRNA and everything is ready before the actual ribosomal complex gets made. So that's what IF3 does. So it has something to do with termination. B would also have to do with termination, so it's not A or B. It definitely wouldn't be E because that's happening at the P site. And D, I'm assuming, is by something different. We good? All right, 33. Which of the following statements is incorrect regarding cis regulators? So this one would be B. Cis regulators do not code for specific gene products. Cis regulators are something that is actually in the DNA. So that would tell us that C is true, because if it's in the DNA, it's definitely part of the gene. And cis regulators are going to be right next to the genes that they're regulating, which would make sense. You'd think they'd be close by if that's the gene they're regulating. And D, they act as binding sites for regulatory proteins. That's also true. So this one would be B. They do not code for specific gene products. All right, 34. An operon related to an important enzyme has a regulator gene that encodes an inactive repressor. So if it's an inactive repressor, it's going to be negative repressible. Yes? I feel like the next one makes it easier to understand. So if the next one's also similar, and this time we have an activator and an inducer, and it was positive inducible. If it's inducible, then it needs an inducer, right? So that tells us that portion of it. And if it's an activator, that means it'll be positive. Does that make sense? Don't beat me up too much. It's been a little bit of time since I've learned this. I remember this being a little confusing when I was in genetics, though. My best advice for you guys for this stuff is, I think I remember there being really good pictures, helping you distinguish between whether it was positive or negative and whether it was an inducer or a repressor. So that would be my advice for this, because I don't think I have a really great way of explaining it to you guys. But that would be the answers for these. All right, 36. Catabolite repression of the lac operon requires, this was going to be camp and cap. So when we're doing this and we're using the lac operon, we do not have glucose, but we have lactose. If we're going to use lactose, the two things we need to start is by having cap bind to camp. Are we good? All right, 37. If a lac operon has both the lac LS and lac OC mutations, RNA polymerase can never bind to the sequence. Is this true or false? False. And I'll be completely honest, I have no idea why. I do not remember what these two mutations are in the lac operon. Anybody happen to know? Okay. What does that have to do with RNA polymerase binding to it, though?
Well, it says can RNA polymerase bind to it, never bind to it, so it can bind to it.
It can bind to it. Okay.
That's what it calls it.
But the shape prevents something else from not binding to it?
The repressor.
The repressor cannot bind to it when it has those mutations? Okay. So in this question, you needed to know that the RNA polymerase can bind. It's the repressor that can't.
Right.
Okay. Okay, cool. I'm always skeptical of questions that say never or always anyways, so. All right, in 38. Which cis-acting sequences in eukaryotes are position and orientation dependent? So the only one in this case are promoters. So both enhancers and silencers are not position and orientation dependent. Does anybody know, are enhancers, like what combination of position and orientation dependent are they? Independent for both? Same thing with silencers?
Yeah.
So these are independent for both. Okay, cool. For both.
If you go back to question 19, that was the p plus q. It's p squared plus q squared equals 1. So that answers why you don't do that. Back to 19.
Is that just another formula that you had?
It's the same, it just, I forgot to add the squares, which is explains why what we
So we could do that without this equation?
yeah, it's just p squared plus q squared equals 1. So that's why you square it.
Okay. So then we found Q was the 700 out of the 2,000. And then we plugged it in here. Because then wouldn't we square it?
It's 0.35 squared.
I thought we took the square root of it.
Yeah, it's 0.35.
So then we found Q squared originally. And we used that. So this was Q squared.
Yeah.
Wait. Yeah, so then we didn't use this equation. We used the P plus Q equals 1, right? Because we found Q squared, and then we took the square root of it and plugged it in here to find P.
I did not do it that way, but you tried.
I mean, I don't know. That's the best understanding I have of this question. I would think they would have to provide you with what proportion were heterozygous to figure that out.
Well, all white is recessive.
Right.
Completely recessive.
But we don't know in the tall ones. Wait, is this tall or red? This is
But
red.
I'm just saying in the p squared plus q squared equals 1 equation, that is specifically just, that's how I did it, because 350 divided by 1,000, which is the total,
Right.
is 0.35. You take the square root of that, that's how you get the 0.59.
Right.
Which is the little r, because q represents the little r.
Mm-hmm.
So then you just, it has to equal 1, so you know the big R is 0.41.
Because these aren't Hardy-Weinberg.
Yeah, these aren't Hardy-Weinberg.
So we can't use the Hardy-Weinberg.
Yeah.
Okay, that makes sense.
You have to use there's another equation where you just solve for a little frequency.
Okay, so it was the equation we were missing then. All right, cool. Okay, that makes sense. All right, so yeah, look for the specification if it's in Hardy-Weinberg or not. Then. Okay, so that's the last question on the practice exam. We've covered them all. Your SI leader said she was going to have 2 more questions. I don't know if she did that or not. But we have a bunch more time. So is there anything specific you guys want me to go through? Do we want to go through like mitosis, meiosis, DNA replication, something like that?
One of the questions is like you have this amount of like 2 and equals 16 and then go through each
Sure.
and how many DNA molecules and
Yeah, let's do that. That sounds good. Do we want to do that for mitosis or meiosis? Yeah. Cool. That works for me. Somebody give me an even number easily divisible by 2. 12, she said it first, so we're doing it. Okay. So our diploid number is 12. So let's say I'm going to have you guys figure out the number of chromosomes and the number of DNA molecules in each phase, and we'll start with mitosis. So, prophase, oh crap, I messed it up. Starting over. What number were we using? 12. Chromosomes, DNA. Let's try this again. Prophase. Metaphase. Anaphase. And I'll say after telophase and cytokinesis, just so we're clear. Okay, so if our diploid number is 12, how many chromosomes are we going to have in prophase? 12. And we know we're replicated in prophase. So what's the total number of DNA molecules we're going to have? 24. So remember what's going on in prophase. All we're doing is, well, our chromatin is condensing into chromosome. And we know we're replicated because we're after S phase. So is anything different in metaphase? No. We're still going to have 12 chromosomes. They're still going to be replicated, so there's still going to be 24 total DNA molecules. But what about an anaphase? What's going on in anaphase? We're pulling all of our chromatids apart. So how many chromosomes do we now have? 24. But how many DNA molecules do we have? Has that number changed? No. All we've done is going from having 12 of those to 24 of these. Because in anaphase, we're pulling those X's apart. And as soon as those X's are apart, each half now is their own chromosome. So these are both a chromosome. So the number of chromosomes has doubled, but we haven't added any more DNA or gotten rid of any DNA, so we still have 24 individual DNA molecules. It's just that they're no longer attached. And then what about after telophase and cytokinesis has happened? How many chromosomes are we going to have per cell? 12 per cell. And how many DNA molecules will we have per cell? 12. Because remember, after telophase and cytokinesis happens, we're going to split half the DNA molecules on one side, half the DNA molecules on the other side, and cut that cell in half. And we also know that since this is mitosis, mitosis is going to be diploid through the whole process. So we know we're going to start with the same number of chromosomes we started with. The only thing we're doing is doubling the chromatids or the DNA molecule so that we can split half in one cell and half in the other. Everybody good with that? We ready to do meiosis? All right, cool. All right. Once again, I'll say our diploid number is 12. So let's go through these. So in prophase 1, how many chromosomes am I going to have? 12, because we always start out with the diploid number. And do we have replicated chromosomes at this point? Yes, because we've gone through S phase, we've doubled our DNA. So how many DNA molecules are we going to have? 24. What about at metaphase 1? Has anything changed? What? Think of, we're starting off with exactly the same thing as in mitosis. We have the diploid number of chromosomes, and we've gone through S phase exactly the same as we did before mitosis. So all we've done in S phase is take all of our DNA and double it.
Oh, it's just that now, instead of them being apart, now you have the mom and dad pairs together.
Right, right. Yes, that's an excellent point. So yeah, keep in mind, we are dealing with homologous pairs in meiosis. So our goal of meiosis 1 is to pull homologous chromosomes apart. And our goal in meiosis 2 is to pull sister chromatids apart.
So for the G1 phase, S and G2 phase, they would all be the same 12 chromosomes?
No, not in G1, because G1 is before S, so we're not replicated yet. So in G1, we're going to have 12 chromosomes and 12 chromatids. Once we get to S phase, we're going to have 12 chromosomes and 24 chromatids or DNA molecules. Does that make sense?
Is that what that would be in mitosis as well?
Yes.
Okay.
Yeah, they both have the same kind of interface thing beforehand. Question?
Could you repeat what you said? You said in the first phase we were trying to pull apart.
In meiosis 1, our goal is to pull apart homologous pairs. So we're pulling apart 2 replicated chromosomes from each other. In meiosis 2, it's going to look exactly like mitosis in that we're pulling sister chromatids apart. You're welcome. If we still have time after this, I could draw that for you guys if you want. We might have time. Okay, so let's continue on. So in metaphase 1, is anything different from prophase 1? Nope. So we're still going to have 12 chromosomes and 24 chromatids. What about in anaphase 1? Is anything different here? Are you sure? It would be the same. This part is where it's a little different from mitosis. So if we pulled apart replicated chromosomes, there's still the same number of chromosomes here. It's different than in mitosis because we didn't pull any chromatids apart, so the number of chromosomes doesn't change. So then what about after telophase 1 and cytokinesis? How many chromosomes will we have per cell? Remember our goal for meiosis 1. 6. Good. We're going to have 6 chromosomes and how many DNA molecules? 12. So keep in mind, at the end of meiosis 1, after cytokinesis has happened, we're going to be haploid. So that means per cell, we're now going to have 6 chromosomes. But those 6 chromosomes are still replicated. They're still doubled. So they're going to be made up of 2 DNA molecules each. So that's why in each cell we have 6 chromosomes or 12 DNA molecules.
Because now the homolog doesn't torn apart.
Yes. Yes. We tore the homologs apart. Exactly.
But you don't now, but it's still one with its replication. That's why you don't have those 2 separate.
Yes. The sister chromatids are still attached, so the number of chromosomes hasn't changed. And if we have time, I'll draw that after we do this. Okay, so at prophase 2, how many chromosomes will we have? 6. And remember, this is per cell. So we have 6 chromosomes and how many DNA molecules? 12. Good. So pretty much we're just picking up with where we left off at the end of meiosis 1. What about at metaphase 2? How many chromosomes do we have? It'd be exactly the same. Good. And then at anaphase 2, how many chromosomes will we have? 12, because this is the part of meiosis where we're actually pulling apart sister chromatids finally. So whenever we pull apart chromatids, that doubles the number of chromosomes. But how many DNA molecules would we have? 12, because we haven't changed that. And then after the whole thing, so after telophase 2 and cytokinesis, how many chromosomes per cell? 6. And how many chromatids per cell, or DNA molecules? It'd be 6. So at the end of meiosis 1, we are haploid, but our chromosomes are still replicated. At the end of meiosis 2, we are haploid, but now our chromosomes are unreplicated. Whenever we have replicated chromosomes, there'll be double the number of DNA molecules per chromosome. And whenever we're unreplicated, the number of chromosomes and DNA molecules is the same. Does everybody follow? I'll give you guys a second to copy that down, and we have a little bit more time. I can draw out the phases. Does that sound good with you guys? If I draw it, might make a little more sense. Is everybody good with this picture? Am I okay to move it? Okay. I didn't hear any no's, so I'll take it as a yes. All right, so I'll just draw through the phases real quick so that we're all clear on everything that's going on. So we'll start with mitosis. And I'm going to keep it nice and simple, and I'm just going to use 2 chromosomes. So at prophase, we're going to have 2 chromosomes. Those chromosomes are replicated, so they're made of 2 DNA molecules each. So this is 1 chromosome, but it is 2 DNA molecules. At metaphase, those chromosomes are lining up at, what is this thing in the middle called? Metaphase plate. So see how nothing has changed here between our chromosome and DNA molecule number. At anaphase, we are pulling apart our chromatids. Our sister chromatids are pulled apart. And as soon as we do that, we now have double the number of chromosomes, because each of these is 1 chromosome and 1 DNA molecule. Does everybody see that? So as soon as the sister chromatids are pulled apart, they are considered their own chromosome now. Let's see if I can zoom in. So after telophase, so after telophase and cytokinesis, this is what we end up with. So we end up with having the diploid number. So we start out with 2 chromosomes, we end up with 2 chromosomes, but they have half the amount of DNA.
So when it comes to all of the transcription, translation, replication, which is pretty much like 23 to 38, you're kind of like shaking on that part, on those parts?
I can help you guys out with transcription and translation. I'm a little shaky on the like operons and that kind of stuff. inducible operons and things like that. Do we want to go through replication, transcription, translation? Yeah, does that sound good? Do you want me to go through meiosis first, or we feel good about that? Up to you guys. I mean, we need to end in about 10-ish minutes. So it's up to you guys what you want me to do. Okay. Let's do the other stuff then. That's fine with me. So do we want to do DNA replication? Yes? All right. So DNA. So this is my double-stranded DNA. So I'm just going to go through and I'll have you guys help me out and tell me all the enzymes we're going to use in the process. Is that cool? So what's the very first enzyme we're going to need to start off our DNA replication process? Helicase. And what does helicase do? Unzips the DNA. So here's my helicase, and it's going in, and as it goes through, it pulls the two DNA strands apart. What do we need to do next? We're going to use our single-stranded binding proteins. And what do our single-stranded binding proteins do? Yes. Keeps strands apart. So I usually draw them as, they're like these little thumbtacks that keep the two strands pinned apart so that they don't come back together. We want to make sure the strands stay apart so that we can access them for replication. So after we have the single-stranded binding proteins, what is the last thing we need to make sure that our DNA is good to go and we can start the actual replication process? What? Gyrase. Good. And what does gyrase do? Yeah, it prevents overwinding. So it's going to be somewhere around here, and it's just going to make sure that when helicase pulls the strands apart, it's not creating tension down the strand. Because remember, although I draw it linear, our DNA is all coiled up because it's a double helix. So now that our DNA is good, separated, stable, what are we going to do first? What enzyme are we going to use next? We're going to use primase. So what does primase do? Adds the RNA primer. So this will be a 5'. So right off the bat, which strand is our leading strand, the top or the bottom? What? Yeah, which one's the leading? The top. Leading strand. Lagging strand. We know the top is the leading because it's going to be making DNA in this direction. Because in this case, 5' to 3' is going this way. If 5' to 3' is going the same direction that helicase is opening the two DNA strands, that makes it the leading strand. Yes. Yes, DNA is only synthesized 5' to 3'. So for the lagging strand, we're going to go this way because in this case, 5' to 3' is going the opposite direction as helicase is opening the DNA strands. So that makes it the lagging strand. So primase adds our RNA primer. And what are we going to use next? Now that we have our primers, what can we do? We can use DNA polymerase 3 to add nucleotides. So DNA polymerase 3 comes in, it finds the primer, and from there, it can synthesize the rest of the DNA strand. But we still have some problems on the lagging strand because we still have a bunch of RNA primers in there, and we need to do something about that. So what enzyme are we going to use to fix our RNA primers? Yeah, DNA polymerase 1. It removes them from the leading strand too. It's just that we have a lot more primers to get rid of in the lagging strand. So it replaces the primers with DNA. So if I had more colors, I would show that, but imagine me removing these RNA primers and replacing those with DNA. Because our end result, we want a whole strand of DNA. We don't want any RNA parts in our DNA strand. That would be bad. So now that we have all of our DNA good, we got rid of all of our RNA, what are we going to do next? What's the last thing we need to do? What enzyme are we going to use? Ligase. And what is ligase going to do? It's going to combine all the Okazaki fragments. Combine Okazaki. So these segments we made the lagging strand in are called Okazaki fragments. And when we use ligase, it's just going to seal them all together, and then we're going to have a continuous strand of DNA. And then we're done. Yay! So really the process is fairly straightforward. It just has a lot of memorization from all these enzymes.
So this is what's happening in bacterial DNA. We don't know what's happening.
Yeah, this is bacterial or prokaryotic.
Because eukaryotic is
Yeah, I think eukaryotic has a lot more depth that we don't usually learn about. Yeah. I don't think this is necessarily just bacterial. I think it's also just anything prokaryotic.
Oh.
Yeah, I know what you meant, but yeah, just to be clear. We don't really have time to go through anything else. Do you have any last minute questions? I think your best bet if you want to revisit transcription and translation would just be follow the diagrams in your textbook. They're actually pretty good. So we don't have time to go through them all, I'm sorry. But any more last minute questions before we wrap up? All right, well, thanks all for coming. I'm sorry Kristina couldn't be here, but hopefully it was still beneficial for you to come, even though I am not a genetics SI leader. But hopefully it was helpful. Thanks for coming, guys.
Thank you.
Yes, sorry.